Welcome to Lecto. Today we are going to discuss active transport in which the molecules move from low concentration to the higher concentration. It is also called uphill movement, and in this transport energy in the form of ATP will use. So here we will discuss the three categories or three types of the active transport. So before we discuss these types, we should know what is carrier protein. Carrier protein has three categories: Uniport, Symport and Antiport. Here you can see this is the cell membrane, or you can say plasma membrane, and here are three different carrier proteins. If we talk about the Uniport, in Uniport carrier protein, one molecule, here is the one molecule move across the carrier protein in one direction. So one molecule start moving across the carrier protein in one direction. It is called the Uniport protein or carrier protein. Next is the Symport, in which the two different molecules move in the same direction. Here are two different molecules. They will move across the protein but in the same direction. So this is called Symport. In Symport, two different molecules move in the same direction. But in Antiport, two different molecules. Here is the molecule 1 and here is the molecule 2. These molecules move but in opposite direction. One molecule will move inside the cell and the other molecule will move outside the cell. This is called the Antiport. So these are basically the three types of the carrier protein: Uniport, Symport and Antiport. Now we will discuss the first of all primary active transport and the secondary active transport. In primary active transport, the molecule will move from low concentration to the higher concentration with the use of ATP. So in that situation, molecule move from low to high concentration. Same is the case, low to high concentration. In primary active transport, only one molecule, keep remember, net one molecule, one molecule will transport. But in secondary active transport, two molecules will transport across the membrane. In primary active transport, ATP is directly used. ATP is used. But in secondary active transport, indirectly ATP is used. So these are basically the difference between the primary active transport and the secondary active transport. Let's discuss its example. Here you can see, let's suppose this is the carrier protein and here is the plasma membrane lipids bilayer. Here is the lipid bilayer. So in that situation, in primary active transport, molecule will move from, if we talk about calcium, so calcium will move from low concentration to the high concentration. Here is the high concentration of calcium inside the cell. So this is move and along with the ATP will also use. With the usage of ATP, the calcium will move from low concentration to the higher concentration. So this is the primary active transport. But the case is sodium potassium pump. So what it is? Actually, sodium potassium pump, if we talk about the sodium potassium pump, here you can see. In that situation, here is the, let's suppose this is the sodium potassium pump, and this is the same here, lipids bilayer. This is the lipid bilayer. In that situation, so concentration of the sodium is low inside, and the potassium which is present outside, the concentration of potassium is low outside. So the sodium will start move from low concentration to the high concentration, because the concentration of the sodium is high outside and the potassium concentration is high inside. So from low concentration it will move to the high concentration, and potassium from low concentration to the higher concentration. Keep remember that 3 sodium will attach to the protein. 3 sodium ions will attach to the protein, and 2 potassium ions will attach to the protein. So 3 sodium will move outside and 2 potassium will move inside. So if we see, net exchange will be 1. 2 potassium, 2 sodium cancel each other. So the net exchange will be from inside to the outside. And due to this basis, the inside of the cell become less positive, or you can say become negative. So this is actually the net transport. So on the basis of this net 1 ion transport, we categorize it in primary active transport. Another is the hydrogen, or you can say hydrogen ion, which is actually present in the kidney. So next is the secondary transport, in which 2 categories fall. First one is the co-transport and second one is the counter-transport. In co-transport, Symport carrier is present. Symport carrier is present. But in counter-transport, Antiport carrier is present. So here you can see, this is the protein, and sodium and glucose will move in the same direction through the Symport. So this is actually called the co-transport, in which the 2 molecules move in the same direction. So example will be sodium glucose and sodium amino acid, and these are actually present in the intestine, through which the amino acid and the glucose absorb in the blood. So next is the counter transport. Here you can see this is the Antiport, and in which the one substance or one ion will move opposite to the other. So here is the sodium and calcium will move in opposite direction. So this is actually the counter transport. And different example like sodium calcium, sodium hydrogen, sodium magnesium, chlorine, bicarbonate, sodium potassium, calcium potassium and chloride sulfates are basically the different types of the counter transport. So next is the special type, in which 2 categories, endocytosis and exocytosis. So endocytosis, in endocytosis a substance will move inside the cell, and in exocytosis substances will move outside the cell. In both situations ATP is used, as we are discussing active transport. Endocytosis has 3 categories: Pinocytosis-Phagocytosis and receptor mediated endocytosis. So here you can see, this is the structure of the cell, and here are the different substances present outside the cell, and here is the nucleus which is present at the center of the cell. So the cell membrane will invaginate inside the, and these substances will be trapped by the cell membrane, and here is the nucleus. And after that, the cell membrane completely invaginate these substances and engulf inside. So here is the, so this is actually the substance which is present outside will move into the cell through by the covering of the plasma membrane. So this is actually the endocytosis. If the liquid substances engulf, then it is called pinocytosis. It is actually also called the cell drinking. If the solid substances engulfed by the cell, then it is called phagocytosis, and it is also called the cell eating. But important thing is receptor mediated endocytosis. This is actually a different thing. Actually, here is the cell and this is the nucleus of the cell. There are pits, there are holes present in the plasma membrane, and these holes are covered by the protein receptor. Here are actually the protein receptor, and these like clathrin. Clathrin is actually a protein which embed in the pits of the holes of the plasma membrane. And the substances which are present outside, if I say these substances ligand, because these are receptors, these ligands will attach to these receptors like this clathrin receptor, which are actually the protein. So after their attachment, these receptors will move inward, leave the plasma membrane and move inside. So they will look like this. Here is the receptor along with their ligand. Here are ligands which are attached to these receptors, and here is the nucleus. So now the ligands or the substances move inward through the receptor. So on the basis of this, we call receptor mediated endocytosis. Endocytosis which happens with the help of receptor. So after that, after finishing its work, these receptors again move towards the plasma membrane pits and fuse in them. So again another receptor will come and attach, and endocytosis will occur. So this is actually the receptor mediated endocytosis. This is most of the clathrin are involved, but caveolin receptor is also involved in the transport of the vitamins. So next is the exocytosis. This is the opposite of the endocytosis. In that situation, here is the cell and here is the nucleus. In that situation, the substances which are engulfed by the cell now will live outside the cell. Here is actually the vesicle, and it will come, here is the vesicle. These vesicle will move towards the plasma membrane, or you can say cell membrane. Here is the nucleus. This is the nucleus. And after that, it will leave out the cell by detaching the plasma membrane. Here you can see this is, it will leave outside the cell, and here is the nucleus, and in that situation the substances will leave outside the cell by detaching its plasma membrane to the cell. So this is actually the exocytosis, in which the substances move from inside to the outside. So this is all about the active transport. If you have any question, then you may ask in the comment section. Thank you.